Hey everyone, and welcome to a deep dive into chemical signaling, especially for all of you in Mr. Martin's ID biology class.
Definitely going to be focusing on stuff you guys are going to see at the higher level.
Absolutely, and we've got all those resources you've been working with, class notes, textbook, chapters, all that good stuff, really centered on C2.1.
Yeah, so think of this as your express lane to mastering how cells talk to each other.
Exactly, and don't forget, you can find helpful diagrams and all kinds of extra resources in the show notes.
Definitely check that stuff out.
So what's really cool about chemical signaling is it's like the basis for pretty much every biological process you can think of. From how your body responds to stress to how bacteria actually coordinate and, do their thing.
Yeah, it's wild.
So let's jump in. And first up, we need to talk about receptors.
All right.
They're kind of like the bouncers at a cell's, exclusive club, right?
Yeah, I like that analogy.
Deciding who gets in and who's left out in the cold.
It's a good one, but it's not just about keeping things organized. I mean, receptor specificity is really important because it makes sure that cells are only responding to the signals that are meant for them.
Right. Otherwise, it'd just be chaos.
Imagine if every signal triggered every response.
It'd be like trying to have a conversation in a room where everyone's just shouting random phrases.
You'd never get your message across.
Absolutely. So these receptors are proteins, and they have these unique binding sites that are a perfect fit for specific signaling molecules.
Called ligands, yeah, and this is that lock and key mechanism.
Right, that ensures that communication is, precise.
Exactly.
And it's not like all the receptors are at the cell membrane, right?
Right.
Some are actually inside the cell.
Yeah, so we have two main types.
Okay.
Transmembrane receptors, so those are embedded in the cell membrane.
Okay.
And then intracellular receptors, which are hanging out in the cytoplasm or the nucleus.
So why the different locations?
Well, it all comes down to the chemical properties of the signaling molecule. So if it's hydrophilic, meaning it likes water, it can't cross that oily cell membrane.
Right.
So its receptor has to be on the outside, like that bouncer checking IDs.
Okay. But what if the signal is hydrophobic? Like a VIP with backstage passes.
Then it can just slip right through the membrane.
Peek right in.
Yeah, and bind to those receptors inside the cell, like estradiol.
Okay.
It's a steroid hormone that does that. It waltzes right in, finds its receptor in the cytoplasm.
So cool.
And then you have something like adenosine receptors.
Okay.
They're transmembrane proteins with this cool 7-helix structure.
Wow.
Weaving through the membrane to catch adenosine molecules outside the cell.
So the receptor's location just depends on whether the signal can enter the cell or if it needs to be detected at the surface.
Right, exactly.
Pretty clever.
Pretty clever.
Now, before we move on to specific signaling pathways, let's take a little detour and talk about bacteria.
Oh.
Because they communicate too.
Yeah, they do. They're surprisingly chatty.
All right, it's like bacteria talking to each other. What?
It's a process called quorum sensing.
Okay.
And they use it to coordinate their actions based on how dense their population is.
So it's like they're taking a head count before they launch an attack.
Exactly. Like, think of it this way. Each bacterium whispers a secret message.
Okay.
And it's a chemical signal called an autoinducer.
Got it.
As the population grows, the concentration of these whispers increases.
Okay.
And when the whispers reach a certain volume, the bacteria know it's time to act as a group.
It's like they're waiting for the perfect moment to strike.
Exactly. And this lets them do things that they couldn't do alone, like form biofilms, for protection.
Right.
Or produce toxins to overwhelm a host.
Do you have a specific example of this in action?
Yeah, one of the most captivating examples is Vibrio fischeri.
Okay.
A marine bacterium that lives in this really cool symbiotic relationship with the Hawaiian bobtail squid.
A glowing bacteria living inside a squid. I'm intrigued.
It is really cool. So the squid gives the bacteria a nice, cozy, nutrient-rich home in a special light organ.
Okay.
And in return, the bacteria's bioluminescence actually helps the squid camouflage and avoid predators.
So it's a win-win.
Total win-win. But how does quorum sensing make the bacteria glow?
Yeah, how does that work?
Well, when the Vibrio fischeri population reaches a certain density inside the squid's light organ, their autoinducers activate a bunch of genes that are responsible for producing luciferase, which is the enzyme that makes them glow.
So they only glow when there are enough of them to actually make a difference for the squid.
Exactly. It's pretty incredible.
It's such a cool partnership, and it demonstrates how even single-celled organisms can play these huge roles in complex ecosystems.
It's wild.
So now let's shift gears a little bit from the bacterial world and talk about how animal cells communicate.
Okay.
It's a bit more complex, right?
A little bit, instead of just whispering in the dark. Imagine a bustling city with different signals for everything. Traffic lights, police radios, ambulance sirens, people calling out to each other.
There's a lot going on.
But it's all coordinated to keep the city functioning.
Okay, I like that.
And in the animal body, we have 4 main categories of signaling molecules.
Okay.
Hormones, neurotransmitters, cytokines, and calcium ions.
That's a lot to keep track of.
Don't worry. We'll break it down. So hormones are like the postal service, carrying messages over long distances through the bloodstream.
Right.
Think about insulin regulating blood sugar levels throughout the body.
Right.
So that's long-distance communication. What about those rapid-fire signals?
Yeah, those are important.
Those are neurotransmitters. They're like the instant messages zipping across synapses between nerve cells.
Okay.
So they're super fast, short range, perfect for things like triggering muscle contractions or transmitting sensory information.
Got it. So hormones are slow and steady. Neurotransmitters are quick and to the point.
Exactly. What about the other 2 players, cytokines and calcium ions?
Yeah, what about those guys?
So cytokines are like the immune system's coordinators.
Yeah.
They're often acting locally to orchestrate responses to infections or inflammation. So think of them as the body's defense force mobilizing troops when needed.
And calcium ions?
Those are like internal messengers, so they relay signals within the cell.
Yeah.
They can change concentrations really rapidly, making them perfect for fast on-off signaling events.
Okay.
Like muscle contractions or triggering the release of neurotransmitters.
So we've got long-distance messengers, rapid-fire communicators, immune system coordinators, and internal messenger.
It's a whole symphony of signals.
It is, and each one plays a critical role in keeping everything in harmony.
Exactly. Now let's delve a bit deeper into some specific examples, starting with epinephrine.
Okay.
The fight-or-flight hormone.
The one that gets us pumped up when we're stressed.
That's the one. Remember those adrenergic receptors we talked about earlier?
Yeah.
Those are G protein-coupled receptors, or GPCRs.
Okay.
They bind to epinephrine.
GPCRs, the ones embedded in the cell membrane with the 7 helices.
Exactly when epinephrine binds to its receptor, it triggers this whole cascade of events inside the cell.
Okay.
The first domino to fall is a protein called a G protein.
And what does the G protein do?
Well, think of it like a messenger relaying the signal from the receptor to other molecules inside the cell.
Okay.
So when epinephrine binds to the receptor, it causes the G protein to swap out a molecule of GDP for GTP.
Interesting.
It's like flipping a switch, activating the G protein, and sending it on its mission.
So the activated G protein is ready to spread the word, but to who?
Its target is an enzyme called adenyl cyclase,
Okay.
a key player in this signaling cascade.
What does adenyl cyclase do?
It's a busy little enzyme. It converts ATP, the cell's energy currency, into cyclic AMP or CAMP.
CAMP. That's the second messenger we were talking about.
Exactly. It's like the megaphone amplifying that signal from epinephrine.
So epinephrine whispers to the receptor. The receptor nudges the G protein, and then TMP shouts to the entire cell.
You got it. And this amplified signal, thanks to CAMP, triggers a whole cascade of effects inside the cell.
Like what kind of effects?
Well, it depends on the cell type.
Okay.
So in liver cells, for example, CAMP activates enzymes that break down glycogen into glucose, releasing that burst of energy into the bloodstream.
That's why we get that sudden energy boost when we're stressed.
Exactly. It's like our body is giving us fuel to fight or flee.
Right. And what about the heart? How does epinephrine affect heart rate?
So in heart muscle cells,
Okay.
CAMP activates an enzyme called protein kinase A, or PKA.
Okay.
PKA is like a molecular switch
Okay.
that can activate other proteins by adding a phosphate group to them,
Got it.
a process called phosphorylation.
So PKA is like a domino hitting other dominoes, activating this whole chain of events?
Precisely. And in the case of the heart, this cascade leads to an increase in the force and rate of contractions, pumping more blood to those muscles, getting us ready for action.
So it's all about prepping our bodies to respond to danger. But this response can't go on forever, right?
Yeah.
How does the signal get turned off?
That's a really crucial point. Signal termination is just as important as signal initiation.
Right, we don't want to be stuck in fight or flight mode all the time.
That would not be very healthy.
Not healthy at all.
So there are several mechanisms in place to shut down the signal.
Okay.
So first, the G protein itself has a built-in timer.
Okay.
It can slowly hydrolyze GTP back to GDP, essentially turning itself off.
It's like a self-destruct mechanism.
In a way, yes. This hydrolysis reaction ensures that the G protein doesn't stay active indefinitely.
Got it. And what about CAMP? How do we get rid of that megaphone once the message has been delivered?
There's a specific enzyme called phosphodiesterase,
Okay.
and it breaks down CAMP, effectively silencing that amplified signal.
So it's like a cleanup crew.
Yeah, removing the messenger once its job is done.
Exactly. And then finally, the epinephrine molecule itself eventually detaches from its receptor, breaking that connection that started this whole cascade.
Exactly. It's like a self-regulating system, making sure that the fight-or-flight response doesn't spiral out of control.
That's remarkable how the body just has these mechanisms to control the duration and intensity of this response.
It really is a testament to the precision and efficiency of cellular communication.
Absolutely. Now let's move on to another important hormone, insulin.
All right.
It plays a critical role in regulating blood sugar levels.
Yeah. Super important.
It's the one that helps our cells take up glucose from the bloodstream.
That's the one. But unlike epinephrine, which is small and hydrophobic, insulin is a peptide hormone.
Okay.
Meaning it's made up of amino acids and can't easily cross the cell membrane.
So it needs a special kind of receptor to get its message across.
Exactly. Insulin binds to the insulin receptor.
Okay.
Which is a type of tyrosine kinase receptor.
Tyrosine kinase, that rings a bell. Doesn't it have something to do with adding phosphate groups to proteins?
Your sharp tyrosine kinases are enzymes that can add phosphate groups to specific amino acids.
Right.
In this case, tyrosine and the insulin receptor is unique because it exists as two identical halves.
Okay.
Called monomers.
Like two puzzle pieces waiting to come together.
Exactly. When insulin binds, it brings these two monomers together, forming a dimer.
Okay.
And this dimerization activates the receptor's tyrosine kinase activity.
So insulin acts as the bridge that connects the two receptor halves. And this connection triggers a cascade of events inside the cell.
You've got it. And one of the key outcomes of this signaling cascade is the movement of glucose transporters to the cell membrane.
Glucose transporters, those are the proteins that act like doorways, letting glucose into the cell, right?
Precisely when insulin signals vesicles containing these glucose transporters, specifically GLUT4, move toward the cell membrane and fuse with it.
So it's like insulin is saying, open up and let the glucose in the cas helps lower blood sugar levels because the cells are taking up more glucose from the bloodstream.
Exactly. And this intricate signaling pathway is crucial for maintaining glucose homeostasis.
Right.
Disruptions in this pathway can lead to problems like insulin resistance and type 2 diabetes.
It's incredible how this complex chain of events is triggered by a single hormone binding to its receptor.
It truly is. And now, before we dive into the specifics of how insulin signaling works, let's take a step back and look at another class of signaling molecules.
Okay.
Steroid hormones.
Steroid hormones, those are the ones involved in growth, development, and reproduction, right? Like testosterone and estrogen.
You got it. And unlike peptide hormones, like insulin steroid hormones have a unique characteristic. They're hydrophobic.
Hydrophobic meaning they don't like water. So can they even interact with the watery environment inside our cells?
That's a great question. Their hydrophobic nature actually allows them to diffuse right through the cell membrane.
Wow, so they don't need any special receptors on the cell surface?
They still need receptors to exert their effects.
Okay.
But these receptors are located inside the cell.
Got it.
Either in the cytoplasm or the nucleus.
So steroid hormones bypass the membrane bouncers and go straight to the control center. What happens when they bind to these intracellular receptors?
Well, these hormone receptor complexes act as transcription factors.
Transcription factors, those are proteins that bind to DNA and regulate gene expression, right?
Precisely by binding to specific DNA sequences.
Yeah.
These complexes can either activate or repress the transcription of certain genes controlling which proteins are made.
So steroid hormones are like master conductors orchestrating which genes are turned on or off.
A brilliant analogy, and their effects are incredibly diverse.
Yeah, they are.
For example, estradiol,
Oh.
a key player in the female reproductive system. It enhances the production of gonadotropin-releasing hormone, or GnRH, in the hypothalamus.
GnRH, that's the hormone that triggers the release of other hormones from the pituitary gland, like luteinizing hormone and follicle-stimulating hormone, right?
You're on fire. And those hormones are essential for ovulation, the release of an egg from the ovary.
So estradiol sets off a chain reaction, ultimately leading to ovulation. It's like a hormonal domino effect. What about progesterone? I know it's also involved in the menstrual cycle.
Progesterone's main role is to prepare and maintain the uterine lining, or endometrium, for a potential pregnancy.
Right.
It stimulates cell growth and makes sure the lining is nice and thick and nutrient-rich, creating a welcoming environment for a fertilized egg.
So estradiol sets the stage, and progesterone gets the house ready for a potential guest.
A perfect way to put it. And if pregnancy doesn't occur, progesterone levels drop,
Right.
leading to the shedding of the lining during menstruation.
It's a beautifully orchestrated cycle controlled by these powerful hormones. What about testosterone?
Testosterone is often associated with male characteristics, but it plays important roles in both males and females.
Okay.
One of its key functions is to activate genes that promote protein synthesis and muscle growth.
So it's like the building blocks for a strong and healthy body. It's incredible how these tiny molecules can have such profound effects on our bodies.
It truly is. Now, before we dive into specific examples of signaling pathways, let's shift our focus to a concept that's crucial for understanding how these pathways are regulated feedback loops.
Feedback loops. I remember Mr. Martin mentioning them, but can you refresh my memory on why they're so important?
Of course, feedback loops are essential for fine-tuning cellular responses to signals,
Yeah.
making sure things don't get out of control.
Right.
It's like a system of checks and balances, making sure the volume isn't too loud or too quiet.
So like a thermostat, making sure the temperature stays within a comfortable range.
Exactly. And there are two main types of feedback loops, positive and negative.
Positive feedback, does that mean the signal gets stronger?
You're on the right track in positive feedback. The response to a signal actually amplifies the original signal,
Okay.
like a snowball rolling down a hill getting bigger and bigger.
So it's a self-reinforcing cycle.
Precisely, a classic example is the estradiol and LH surge during the menstrual cycle.
Right.
As estradiol levels rise, they trigger the release of more GnRH and LH, which further increases estradiol production.
So it's like a hormonal chain reaction,
Mm-hmm.
pushing estradiol levels higher and higher, ultimately leading to ovulation.
Exactly. Positive feedback is all about pushing a process to completion. Now let's contrast that with negative feedback, which is all about maintaining balance.
Okay, so if positive feedback is like a snowball rolling down a hill, what's a good analogy for negative feedback?
Imagine a thermostat maintaining a comfortable room temperature.
Okay.
When the temperature gets too high, the thermostat kicks in to cool things down.
Right.
When it gets too low, it turns on the heater to warm things up.
So it's constantly adjusting to keep things stable.
Exactly. And negative feedback is crucial for maintaining homeostasis.
Okay.
For example, as testosterone levels rise, they signal the hypothalamus and pituitary gland to reduce the production of hormones that stimulate testosterone synthesis.
So high testosterone levels actually trigger a response that lowers testosterone production. It's a self-regulating system.
Precisely. And another great example is the regulation of antidiuretic hormone, or ADH.
Okay.
When the body is dehydrated, ADH is released to promote water reabsorption in the kidneys,
Okay.
increasing blood volume.
So ADH helps us conserve water when we need it.
Exactly. And as blood volume increases, ADH secretion is inhibited, preventing us from retaining too much water.
It's like a delicate balancing act, keeping our water levels just right.
Precisely, these feedback loops are essential for maintaining homeostasis and ensuring our bodies function properly.
They're all so fascinating. It's amazing how much complexity is hidden within these seemingly simple processes.
It truly is, and we've only just scratched the surface of the intricate world of cell signaling.
I can't wait to learn more, but before we delve further, let's take a moment to recap what we've covered about feedback loops.
Great idea. So feedback loops are mechanisms that regulate cell signaling pathways, ensuring that responses are appropriate and balanced.
Right, there are two main types: positive feedback, which amplifies the signal, and negative feedback, which restores balance.
And these loops are crucial for maintaining homeostasis, preventing signals from getting out of control. It's wild to think that just one tiny signaling molecule can set off this huge chain reaction, leading to all these effects that amplify the original signal.
Yeah, it shows how powerful and efficient these communication systems are. But it's not just about amplifying the signal, right?
Right.
It's also about controlling it, making sure the response makes sense and that it doesn't go on forever.
Yeah, that's where those feedback loops come in.
Right, they're like the brakes.
Exactly. They keep the signal from spiraling out of control.
And we talked about both positive and negative feedback loops, right?
Right.
Each playing this crucial role in keeping everything balanced.
It's like all these different parts, the receptors, signaling pathways, second messengers, feedback loops,
Yeah.
all working together like this perfectly choreographed dance.
Like a symphony.
Exactly. Each instrument playing its part to create this beautiful, harmonious sound.
And just like a symphony, if one tiny thing is off,
Yeah,
it can mess up the whole piece.
that's a great analogy. Yeah. Disruptions in any of these components can lead to problems, from small imbalances to really serious diseases.
Right, like cancer, diabetes, autoimmune disorders, all those things we talked about.
Exactly. Understanding these pathways and how they can go wrong is so important for developing new treatments.
It's like trying to find a broken wire in some super complicated circuit.
Exactly. By targeting specific parts of these pathways, we can maybe develop drugs to block or enhance signaling depending on what we're trying to do.
This deep dive has been so eye-opening. It's amazing how complex and important these processes are.
It's really a fascinating field, and we've only just scratched the surface. But for all of you in Mr. Martin's class, I hope this deep dive has given you a good foundation for understanding chemical signaling.
For sure. And remember, guys, this is just the beginning.
Yeah, keep digging into your textbooks, review those notes, and don't be afraid to ask Mr. Martin any questions you have.
Absolutely. And speaking of exploring further, don't forget to check out the show notes for this episode. We've got links to some helpful resources and diagrams that might help you visualize things better.
Yeah, definitely check those out.
And if you found this deep dive helpful, please like and subscribe to the deep dive.
Yes, do that.
Sharing with your classmates is a great way to help each other out and, succeed in IB biology.
Definitely. Thanks for joining us on this journey into the world of chemical signaling.
Until next time, keep those brains buzzing.